Noble Capital. Our next presenting company is Cocrystal Pharma, a company developing antivirals for serious diseases and infectious diseases. With us is Jim Martin, the co-CEO. Mr. Martin, please go ahead.
Great. Thank you. Thank you, Robert. Thank you to everyone watching this for your interest in Cocrystal. And of course, thank you to Noble. Always a pleasure to be a part of this. So you guys should see our deck up on the screen. It's a pretty short meeting here, so I'm going to run through it pretty quickly. But there are references to our to contact us if you guys have any follow-up questions, whoever's looking at this. So forward-looking statement. So Cocrystal is a therapeutic company. I always like to make that clear. We're not a vaccine company. Vaccines generally are more preventative. Therapeutics are when you get sick to get you better. And that's what we do. And we focus on primarily three areas of influenza, norovirus, and coronaviruses. And we have a drug discovery platform that comes off of one of our founders, who's our chairman, Dr. Roger Kornberg. What he took his Nobel Prize and took that technology and industrialized it to be able to have this machine that can really create these antivirals. And the way we do it is to create very broad spectrum antivirals, and I'll talk about that a little bit further on how we do that through crystallization process, and also very potent. So highlights of the company is that we do go after multi-billion-dollar markets for these viral diseases. We do have a very proven drug discovery platform, which is used to develop all of these drugs. These are all 100% owned that we have created. There's no licensing in here. These are all, whether it's they're all clinical stage. And we have a seasoned leadership team. We have a couple Nobel laureates as part of our team here, and we have a very cost-efficient operations, no debt. So the full pipeline includes multiple influenza assets that we look to move forward. We also have norovirus and COVID-19. Now that's interesting there, and that you'll see there's the drug's name is CDI-988. That's our internal name for it right now, and it's for both indications, and that's already completed a phase 1. So this is a very potent drug that has multiple applications on it, and we also have additional respiratory diseases. The way we create these drugs, going back to that platform I spoke about, is we have a crystallization process where I'll summarize it at a high level for you here, is that we can create a crystal out of the virus itself, sort of like a salt crystal, and they shoot these high X-ray beams through it, and it's like lights up the atomic level structure of the virus. And what we do that's very important is we'll take every strain of the virus, because just pausing for a second, viruses have two strengths. One is they replicate incredibly fast as a disease, the virus, and then the second thing is they mutate. So we want to make sure we take every family member, in a sense, we look for this one area that, and these family members, if you take influenza, there's a whole bunch of different strains. Those strains are just mutations that have happened over time. And some of them become prevalent, they become strains. And so what you want to do is you want to look for this one area that no matter how many times and how many strains you have, hasn't changed in that replication area. Because if it hasn't changed, or if it ever did change, it never carried on. It killed off the virus. This area is what you call an essential area. We're looking for that essential area no matter what strain is required. And that's where we bind compounds to, and we disrupt the replication. A long explanation for how we go about creating these drugs. And the bottom line is we create very potent and very broad spectrum across the whole family is what we're looking at to create drugs. The norovirus program, we'll jump into that. So norovirus, in case hopefully some of you have heard of this, it's a pretty nasty little virus. It makes the news a lot because it takes cruise ships, and they'll turn around and have to come back home if somebody gets sick, because this is a very rapid, fast spreading disease, and it will just shut down a cruise ship. And so it has a lot of military issues, schools, nursing home, and restaurants. What's interesting, the opportunity here is that there is no vaccine, no therapeutic approved in the world for this drug. I mean, for this disease, there's just nothing out there. They just treat the symptoms. So it's wide open for someone to come in like us with this fantastic drug and help out. I mean, we have a $60 billion annual burden on the world. There's, it's just a real problem, this norovirus. This slide here is showing that we have this is where we attach, we bind. This is the actual replication process of the virus. And what we do, as I said earlier, we bind and we stop that replication. And we bind to an area that's unique, and the same no matter what strain of the virus there is. Recently, we've had outbreaks. We continue to have outbreaks. Norovirus isn't going anywhere, and we've had, we've had a lot of news lately and a lot of spreading of it. It's the Ferrari of viruses. It really does spread very quickly. Here, this slide kind of speaks to the different strains of the virus. So you have different family members, but yet we find this one area that's the same in all of them, and that's where we bind to, and we disrupt that replication. We've already completed phase 1 on this drug 988. The phase 1, the indication was for both coronavirus and norovirus. Just so you know that you can do this when you have a phase 1, because it's for safety and tolerability. It's not efficacy. So we have both indications. We took this drug into single and multiple ascending doses. It's very tolerable, very safe. I mean, this phase 1, the biggest issue that a lot of companies run into is tox. If you have a toxic compound, it looks great, it works great, but it's toxic to humans, then you got a problem. So we put it in humans. We went from 100 to 1200. It's been very safe, very tolerable, all the way up to 1200 milligrams. So we have passed that with flying colors, and now are moving forward. Here's just some more data on that trial. Headaches is typical because when you're in a trial, you have to stop smoking and drinking coffee. A lot of people get headaches, but it went good. So now we're moving forward. We're going to do what's called a challenge model. We're going to take the healthy patients. We're planning to have 3 arms on this. One, you're going to have a placebo, of course, and then we're going to actually take people. The way these trials work, you'll take a subject, you infect them with the virus, and then you'll make sure the virus takes, and then you'll give them the treatment. But what we're also doing is we're going to give people the treatment first and then infect them with the virus. And this is to check the prophylaxis or the preventiveness of this virus. Because if you're on a military ship and you get sick, this thing spreads, a Ferrari of viruses. I mean, the way it spreads, it's really fast. You want to be able to give the sick people the drug, and people that aren't sick the drug to prevent them from getting, sick. So that's why we're conducting this. Data is everything in the markets these days, and we want to have some data to show the effectiveness of this drug, not only as a therapeutic, but we'd love to show its preventative aspects. And the government is very interested in this, very supportive, especially the military. The cruise industry is very supportive of this, and we just need to get some data so we're moving that one forward. Pandemic, influenza. I mean, we all have had influenza. You get the vaccine, you still get influenza. I mean, that's no fault of the vaccines. I mean, vaccines are a tricky thing. You have to kind of predict the prevalent strain, and because you have to manufacture a lot of vaccine and give it to people, and if it's a different strain, well, you get sick. So but therapeutics are a little bit different because, in the way we create our drugs, no matter which strain it is, we're binding to an area, stop that replication through all these various strains. So it's a huge market. There are some therapeutics like Tamiflu out there that's been out there for years. It's already been mutated around. A new one that's come on. There's Tamiflu, and then there's Ofluza, Japan, and they are already seeing mutations work around that. And that's why we developed these in this one highly conserved area using this platform that we use to identify that at the atomic level structure, so that we can hit an area that it cannot mutate around. We realize how fast these viruses can mutate. So we've already completed a phase 1. We have a phase 2 that's ongoing. We've had some issues with that. So it's a bit delayed here because there was an outbreak of the influenza at the time we were conducting the trial. So we're either going to continue with that CRO or move on to a different one. But that's an ongoing trial. But it has completed its phase 1, and everything is looking really good. And what this slide shows here is an example of all the different strains of influenza that we have targeted with our drug. And the nanomolar at the top are very low. It shows the high potency of our drug. And we designed this drug for all of these. So, this is very important. The mutation of this disease. And just starting last year, there's been some concern because everyone probably already knows that influenza can go from humans, and then it started going to birds, and you got bird flu. More recently, around last year, and you can Google this and look it up, but there's, it's now going to cattle and bovine. So it's not a lot, but it's in here in the U.S., and it's very concerning on what's going to go on with that. So it's the need for a very potent antiviral drug is important, and we have the government. We've actually hired a consulting firm to help us about 2 years ago. Prior to that, we were trying to, we were working ourselves, but the government is you almost have to have a full-time group on that. It's a lot involved with getting these government contracts. So we brought in a group and put in applications, and it takes about almost a year on some of these. So we're looking to get those answers on those that we had put in. Everything's looking good. Probably won't get, might have some things coming in this year, but next year and the early first quarter is when we're expecting to get, well, unless this government shutdown affects it, we were expecting to get some answers back early in Q1, worst Q2 next year on some of the larger applications that we put in for these grants and these fundings. So they're looking positive, but so we're involved with that, trying to move this into the area of some more additional government funding for some of our programs. This slide really shows how we bind to different strains. I mean, we're really emphasizing it because it really is a strain that is just almost required if you want to really have a real good drug for this disease. So here you have the H1N1, the H5N1, the H7N9. These are all strains of the influenza. And what you'll notice is that, and this is like an atomic level graphic, where you'll see at the very top, H432 is the same in all of them. To the left of it, F325, meaning no matter what strain you have, this is an area that has, in the replication area, that it's the same no matter what strain, and that stops the replication of this drug by binding to it. So we have this shows just the broad spectrum side and how we develop these drugs, and this is our sort of our secret sauce on going to have the what we hope to be the most successful drug for a treatment of influenza, which continues to be a problem. That's what this slide speaks to. I mean, there's still, as probably most people understand, influenza has not gone away. Now, every drug I spoke about, whether it's the noro coronavirus or the influenza, it's all oral, is the way we developed it. But we also took the same influenza drug, the 42344, which everything I showed you was for the oral pill, but we've also formulated that for inhalation. And we did some testing, and what we did is we tested with human lungs. And that's what this slide speaks to. So there's really not much to say on this slide. I think it speaks for itself, as you can see, with and without treatment. The epithelia or the shag carpet looking lung is really needed to be able to work. That needs to be able to grab the oxygen, transfer it. And you can see all the damage that influenza can do when you don't have a treatment. So we're really excited about this. This is still preclinical, this formulation. We have to, we're looking to further develop that into clinical stage. We do have government interest that we're working on, and so hopefully you'll get some news on that soon. Again, it's already been cleared on the phase 1 and currently in a phase 2. We have a very strong board. Dr. Roger Kornberg at the very top there, he is one of the founders. He's the Nobel Prize winner. He's the one that worked to industrialize the process to create this platform. The company was originally funded by Dr. Philip Frost, who's been very successful. Fred Hassan also. So the slide here shows we have a very strong biotech board here. This isn't a group that doesn't know what they're doing when it comes to developing drugs and working in this industry. So there's no amateur hour here with this group here. So we have a very strong board. To the left there, you'll see management. That's Sam and myself, Dr. Lee. Dr. Lee is also one of the founders, along with Dr. Roger Kornberg and Philip Frost. Our scientific advisory board we have there has 2 Nobel laureates, very strong a scientific company, very strong into science. And that's, we do a lot of work before we go into clinical trials, even in phase 1. There's a lot of cost involved with clinical trials, even though we've done really good with the cost, especially working in Australia. The company makes sure that we do all of the data and the clinical work ahead of time to make sure we have the highest chance of success in the clinical trials. And with that said, on the business side, we make sure we patent and protect everything we do. That board I showed you earlier, they have had success, and with success comes challenges, and you really want to make sure you patent and protect every little thing you do. Next steps, the norovirus, we're going to do that challenge model I talked to you about that has the 3 arms, the one where you have the preventative side, the treatment side. And also with 42344, we are going to continue that trial to get our results on that phase 2 trial. Company itself, 4.8 million as of June 30th. Recently, we raised another 4.7 million with 8.3 million potential from warrants in the future. Those are, by the way, those are very clean warrants, very, very plain vanilla, no ratchets, there's no cashless exercise, there's no repricing. They're just very plain vanilla warrants. The common shares outstanding and diluted, that's current. That isn't as of June 30th. That's the current. And, we have some decent liquidity in the stock. So just to close out here, the highlights again, I showed this to you earlier briefly. These are multi-billion-dollar markets. We have a very strong proven platform for developing these drugs. We make a very broad spectrum, very potent. The broad spectrum is very unique to us in that we're looking to develop these things to treat all of the strains of the virus. We have a very strong board, a very strong scientific team that includes the Nobel laureates, and we run a very cost-efficient operation with no debt. And that concludes, the presentation. I'm going to see if Robert, maybe turn it over to you, see if you have any questions.
Yes, thank you, Jim. Just in viewing all the slides and all the science about how these drugs work, would I be correct in thinking that your therapeutics bind to highly conserved areas of the enzyme that are carried forward on all strains before they develop into strains, so it can work on the early steps of the virus replication, and it is a universal therapeutic?
Yes. Trying to back up the I'm not going to find it, but yes. So as I said, we are attaching to an early part of the replication area. We bind our drug to an area that is the same no matter which strain we have tested. We want to make these broad spectrum drugs. Now, we also, it's conserved region. I also like to say another way of saying it is it's an essential region. In other words, it's never mutated this area. I mean, it has not, because it cannot live if it mutates this area. So by binding to that area and disrupting that process, that's how we're able to create these broad spectrum drugs. And um, some, not trying to get it too much ahead of myself, but some of these areas are very similar in different diseases. And that's how we're able to create a drug like CDI 988 for norovirus that also works in coronavirus. And when I say coronaviruses, I didn't say, COVID-19. I didn't say Middle Eastern respiratory disease. I say coronaviruses because it's all of those that fall under coronavirus. COVID-19 is a coronavirus. So we're not just going after COVID-19. We go after the full uh, gamut of these these strains. So yes, we bind to these essential areas that have not been able to mutate historically. And that's because if you mutate that area, it doesn't seem to exist. So it can't live. So, and that's where we bind. And that's all comes off of our drug discovery platform, which really gets us down to that atomic level structure of the virus and allows us to be able to locate those areas.
Sure, that's an important point. Binding early in the replication cycle before the full virus develops and has strains and going after the mature virus. But why did you select the norovirus as the initial indication for the panviral CDI 988?
Yeah, no, you have to make decisions. It's a business decision. Um, we don't have the money to just go after both indications. These trials, they cost money, and there's a lot of people, a lot of players out there in the coronavirus area ever since COVID-19. It was so we had to make a decision. We decided norovirus because first of all, as I said, there's no vaccine or therapeutic, so the market's yours for the taking if you can come up with a drug. It is a prevalent disease with a large potential with the military, and with this disease and with the coming up with a drug, there's a uh, very large market for stockpiling of the drug. When you come up with an approved drug, the stockpiling is going to be huge because the military, the cruise lines, everywhere will just stockpile this drug because when a person gets sick, you need to, it's usually large groups of people, and then you have to quickly get that out to them. Um, so that's where if just as a business person, the real money will be also in that stockpiling. So just in summary, there's nothing for this disease. It's wide open. So we decided to go with this first. Not that we don't plan to we still plan to take it forward with the coronavirus.
Well, yes, as you mentioned in the slides, norovirus is most closely identified with cruise ship illness, but is very prevalent anytime you get large numbers of people living together. Well, whether it's in the military, nursing homes, and all of the other places that you mentioned in the slides. So why is it important to develop it as a therapeutic and a preventive?
Well, we'll just use the ship, whether it's the military or cruise ship, or whether it's a nursing home or whatever. We have all these people kind of stuck in this area. You're not going to, nursing home, it's not like, okay, everyone go home. It's not like a convention. So we have these group of people stuck in an area. Um, and the key here is this is the Ferrari of spreadability viruses. I mean, it really spreads quickly. Um, that's just it's inherent fact on that disease norovirus. And so you want to be able to treat people and prevent the people that are stuck either on a ship or in a nursing home or on a cruise. So you really want to be able to treat and prevent everyone else from getting sick because this disease spreads very quickly in multiple ways. You can touch things. So the idea here is if you could treat and prevent, that will be the key. And that's
Great.
the
And
goal we're setting.
speaking of treating and preventing, why are you pursuing a human challenge study as the next step in development? And can you tell us a little bit about the study?
Sure. So the way I like to think of it to explain it to people is that, influenza is all over the place. It's constantly popping up. It's seasonal. Norovirus is, there was just recently a ship from California that just docked here in Miami that had norovirus. It pops up and it's very, it's usually out on a ship somewhere. It's not as efficient to try to chase these things down and run a trial. And you're not going to have a hospital to do a trial on a cruise ship. So the challenge model is the most efficient and effective way of one, making sure you control the infection, making sure you control the ability to see if there's a prophylaxis or preventative side, because you're actually starting off with healthy subjects, healthy volunteers that will, you'll actually document giving them disease. You'll document the disease takes effect. You'll document the treatment, and it also makes things much quicker to get this through this trial, because you're gonna you start off with healthy volunteers and you go through the cycle. It's not like you're waiting or chasing a group. So it's efficient. Cost wise, it's efficient. Scientifically, it's efficient. The data is very good, and the trials are going to be shorter. So the tricky part was just finding the disease, and we've been all over the world, and we've the logistics of getting this trial together was probably the bigger challenge with norovirus. So, and we believe, and we've got that going now. So we're going to start that this year and look to have data out early next year.
Okay, great. It sounds like you're controlling all of the variables that would lead to variations in the results. So it sounds like a very good, efficient trial. So one other thing is about the trial and other companies developing norovirus. What gives you confidence in your approach?
There's not a lot of companies that have had success in developing this. I think we've had success because of our crystallization platform, our ability to get to that atomic level structure of the virus and identify these key areas in the different strains of the virus, in our ability to find a way to bind to that area. So I'm going to just say it's the uniqueness of our process for developing drugs off of our platform that we think, and we preclinical testing is very positive. Our phase 1 has shown, good results. And
Great.
data is key here. We have to have data. So we're gonna have some data early next year on this.
Okay, and just switching to CC42344 in influenza. Can you tell us the status of the program?
Yeah, so I mentioned earlier we're in a phase 2 on that. We're having some issues with the CRO over in the UK. We might be moving this over to Australia because the prevalence of the disease and finding it isn't coming out with